This is an OCR A-level biology video on the cell cycle unit 2.6. I think I'm going to split these videos up into cell cycle, mitosis and meiosis, and then there'll be a separate video on the specialization of cells. But first of all, in this video, let's look at the cell cycle. So we're going to look at interphase. So in interphase, we're basically looking at three main stages: G1, S and G2. G0 is also included in this, but on the whole in interphase the cells are growing and preparing themselves for M phase, which is the mitosis and cytokinesis stage. So in G1, which is the first one after mitosis, the cells grow and increase in size. They undergo transcription, organelles duplicate, they have biosynthesis of enzymes needed in the S phase, and the whole process is controlled by this p53 tumour suppressor gene. Within G1 we've got this G1 checkpoint over here, and these checkpoints come about at various stages in our cycle. And for the G1 checkpoint, it's just checking that all the chemicals needed to enter S phase, which is the second phase where the DNA replicates, are present. And it's just checking that there's no damage to the DNA, because in S phase we're going to copy our DNA, and if you've got damaged DNA then the cell could be mutated and you can get cancer forms. So p53 tumour suppressor genes, that's dealing with cancer. If you can see the links, we don't want cancerous cells to be produced. So if it gets past this G1 checkpoint, which if it fails, by the way, it will go to G0, which I'll speak to you at the end. So if it gets past this checkpoint, we go into S phase. So in S phase we have unwound chromosomes. Every molecule of DNA is replicated, but first of all the housekeeping genes are replicated. So the housekeeping genes are the sort of active genes, essential genes to be within the cell. They're replicated first, then the inactive genes are replicated last. It's a rapid phase because as the DNA is exposed, it needs to be as fast as possible to prevent possible agents or mutagens affecting it and destroying the DNA, or just causing mutations really. And finally in the S phase we have sister chromatids produced. One more thing that does happen in S phase is duplication of our centrioles. The book I have for this, OCR biology, says that the duplication of centrioles, the centrioles split, or centrosomes split, during the prophase of mitosis, but doing a little bit more research, they actually duplicate in S phase and sort of in G1 phase, and it depends on the cell. So one thing to think about in the whole process is our aim here is to double the amount of DNA we have and to double the amount of organelles we have, because then we split the cell in two basically, and one gets full amount of DNA and the other gets full amount of DNA, and they all get equal share of organelles, and that includes centrioles. So whatever source it tells you, centrioles at some point during interphase, depending on the cell, it can be G1S, I don't know if it can be G2, at some point the centrioles will divide and they will split during prophase. So G2 is when the cell just keeps growing and proteins are stimulated for mitotic cell division. So we have this final G2/M checkpoint to check that all the DNA is replicated. So that's a really essential checkpoint. If not everything's correct at this checkpoint, then we can enter G0 again, the cell will become inactive, or they can just fix the errors. Right, so what is G0? It is the phase that is not included on this diagram, but it's the resting phase. So some cells go straight from M phase to G0, and cells such as neurons enter G0 phase because they just want to carry out their function. They don't need to enter the cell cycle yet, so they enter this G0 phase. G0 phase can also be a phase for cells which are old. Cells usually have about 25 to 30 cycles, and they enter this G0 phase pre-death, pre-apoptosis, just to get out of the cell cycle, because that DNA has been replicated so many times it increases the chances of mutations which we do not want. So that is the cell cycle. It's quite easy to understand. The final thing is a metaphase checkpoint, but that's more to do with mitosis. It's in the middle of mitosis. One final thing I'm going to do in this page is sort of draw out what is happening to our cells within the cell cycle. So first of all, we've got this normal cell here. So in interphase, we've got, by the way, the whole of this video and future videos, I'm using two chromosomes, one orange, one green here. There are actually 23 pairs of chromosomes, okay, but I'm just doing two of them because it would be too complicated to draw them all out. It just makes it easy to represent. Okay, so at this point, chromosomes are in their chromatin form. They're loosely found within the nucleus, and this is our centriole or centrosome. So then we have G1. So in G1, the cell grows, which we'll show to you in a second. We have the same amount of DNA here, but look, the cell's just getting much larger. I've not drawn the other organelles, but you know they'll be duplicating as well. I've spoken to you about it here. Just keep refreshing those points. I've not drawn everything on this diagram, okay? Then we have S phase. So we have that DNA, which was, so two chromosomes, this large cell now. And what I'm going to draw out here is what's actually happening. So we call this one strand here a chromosome, and we call this, which is two of these sort of strands, a chromosome as well. So both of these are chromosomes. That is why so many people get confused in this topic, I think, because we're using the same name for one as we are for two. They are both called chromosomes. So if you ever get confused, people saying, oh, there's one, so that's a chromatid. No, when it's on its own, not in this formation, it is a chromosome. Two together also make a chromosome when they've got a centromere in between them, binding them together. But these two strands are then called sister chromatids. Okay, so a chromosome, two sister chromatids make another chromosome in this, but both of them are called a chromosome, even though when that is put in that, it will be called a sister chromatid on its own. Okay, that's where we get lots of issues in this topic. Personally, I find it too vague, and I think they should have separate words for the two molecules, but there must be a good reason for it. Finally, we have G2. So the cell keeps growing, and I've tried to draw on the cell bigger. It's not that noticeable, but that cell is bigger. We've got these molecules of DNA which have been replicated now. So there was two originally. They're both replicated, and then we've got four. As you notice here, our centriole is also divided, and if you ever get confused about centrioles and centrosomes, so centrosomes are the orange bit, centrioles are these two sort of tubular structures inside our centrosome, but I've noticed in the textbooks it ignores centrosomes and just calls them centrioles, so I think in our specification we could get away with saying that, talking about centrioles rather than centrosomes, but I'm just going to teach you both So that was the cell cycle unit 2.6. I'm going to split this down into 2 or 3 videos. In the next video we're going to look at mitosis and maybe meiosis in the same video, depending on how long mitosis takes. Alright, thank you for watching.